Okay, folks, wanted to do a recording that went into a lot of detail since I know this was a lot to learn at once in class. So let's do some quick review real quick before going into the homework itself. Up here on the toolbar, we have A for action, S for show, H for hide, L for label, and C for color. In question 1, I ask you to convert the structure of hexokinase to surface mode. Is hexokinase in the open conformation or closed? You might want to reference the hexokinase slides in glycolysis 1. In those slides, I kind of show you, the structure with the open lobes or the closed lobes. So let's do it. To go to surface mode, we'll go up here to S. We'll click Show. We'll click Surface. And you'll notice that once I do that, you can kind of see this like space filling mode where we know the general shape of hexokinase. Here's one lobe, here's the second lobe. And you can see this kind of open-mouthed Pac-Man structure. When I rotate this molecule and zoom in a little bit, you might notice that this molecule, ADP, is exposed to water. Look at these phosphates just kind of hanging out, totally exposed to water. Now, using what we see in terms of the overall structure and the fact that the substrates are exposed to water, we should come to the conclusion that hexokinase is in the open conformation. Next up in question 2, I want us to kind of come to a conclusion about what could possibly be the base that does the catalysis in the mechanism of hexokinase. In the lectures, I just gave you a generalized base B, but let's do some exploring of the structure and see if we can determine if there is a plausible base near our glucose molecule. So what I want to do is first hide the surface. So let's go to Hide, Surface. And I want to zoom in on that glucose molecule itself. Let's actually even make this a little bit bigger. Here we go. So here's our beautiful glucose molecule. Here is our ADP, although we know in the mechanism itself it should be ATP. When we are trying to find amino acids nearby, it's best if we actually show the primary sequence. On the bottom right here, folks, there's a button that says S. This S means sequence. When I press that S button, you'll see that up at the top here, let me move my face real quick. Okay, hopefully that helped. Up at the top here, you'll see that the primary sequence is shown. Additionally, you'll notice that there are some numbers. Each of these numbers directly corresponds with the amino acid below it. So here at 571, the letter right below the 5 is amino acid 571. This amino acid is G. If we remember our one-letter abbreviations, we should remember that glycine is G. So glycine at 571. Now we're ready to start question 2. Find all residues within 5 angstroms of the glucose molecule. Which would be most likely to perform base catalysis? I have some instructions here on how to do it. You can follow along. So if we look at the mechanism of hexokinase, we'll see that the 6' OH is deprotonated, so it can perform a nucleophilic attack on ATP. So that means we need to be finding residues near this hydroxyl because this is the one that's going to be deprotonated. Let's do that. First, click on the glucose molecule. Then next to selection here, go to action. So action, scroll down to modify, go over to around, and let's find all residues within 5 angstroms of the glucose molecule. And click it. At this point, you see my selection has changed. Look at all of these pink dots. Those are the amino acids nearby. So let's show the sticks so we can actually visualize them. So go to Selection, Show, Sticks. And you'll see that now we can see all the stick structures. That makes things a little bit more difficult to see. So I'm going to reorient us and get us a little bit closer. Here's my glucose molecule. Here's the hydroxyl that needs to be deprotonated. We could perform a quick visual assessment and ask ourselves, okay, what amino acids are really close by? I see 1, 2, 3 amino acids that are really in close proximity to that hydroxyl group. You could solve this by finding those 3 residues and asking yourself, can they do acid-base catalysis? You'll notice that when I click on the residues, they're highlighted up at the top. One of them is a serine at 60123, so serine 603. We have a lysine at 621, and we have an aspartate at 657. So those are the 3 closest residues. Serine does not have a pKr, so it's not likely to do base catalysis. Both lysine and aspartate have pKrs. They could be base catalysts. Additionally, look at how close they are to that hydroxyl group. They're super close. So they're in close proximity. They seem reasonable. So lysine 621 and aspartate 657 seem plausible. Now let's see if either of these last 2, glutamate 708 or 742, are likely candidates. To find these, I'm going to go to the top here. Let's scroll over to the 700s. We're looking for 708 and 742. So here's 708 and here's 742. I'll reorient us real quick. Here's the hydroxyl we need to deprotonate. Well, 1 glutamate's over here, kind of on the opposite end of the glucose. That does not seem like a good choice. The other is way over here. And, you know, it's certainly in a better position, but I don't really think it's very close to this hydroxyl either. So I'm going to say neither of those are good candidates for base catalysis. And so it's probably only the lysine and aspartate that could possibly do this catalysis. Let's move on to questions 3 and 4. In order to do questions 3 or 4, I'm going to quickly make my screen a bit more simplified. So I'm going to hide all of the sticks that we just showed. Okay. I'm going to zoom out a bit. I'm going to reorient us to this ADP molecule. So here's our ADP. The question's asking us to find amino acids around ADP now. So using the same method, let's find all residues within 5 angstroms. Click on the ADP. And we'll say Action, Modify, Around, Residues within 5 angstroms, and click it. You see a bunch of new residues are now selected. Let's show them as sticks. Show, Sticks. And now you see we have a bunch of new amino acids popping out here, right? Here's a little phenylalanine hanging out at the bottom. A bunch of other amino acids are being shown. Question 3 says name an R group that is performing ion dipole interactions with the phosphates of ADP. please use this format. Question 4. Name an R group that's performing ionic interactions with the phosphates. Let's do it. So maybe we should first kind of familiarize ourselves with what's going on here. Here's our ADP molecule. If we recall nucleic acid or nucleotide structure, there's a base, there's a sugar ring, and then there's phosphates at the end. This is ADP, so we have 2 phosphates. The question is asking about the phosphates, so let's zoom in there and kind of just focus. We need to find an amino acid that does ion dipole first. So that means since the phosphates are charged, we need to find a residue that's polar but neutral. Let's take a look around. Here's an R group coming off. It looks like a methyl group. That should be alanine. That seems like an unlikely candidate. It's not polar. Here's one up here. You can tell by the structure what the R group is, or if you're unsure, you can scroll to determine that this is an asparagine. Asparagine is neutral. Is it in close enough proximity to do ion dipole? What else do we have around here? Well, I'm seeing a couple more residues here. What's this? This looks like a threonine. Yep, we have a threonine here at 680. Is it in close enough proximity to be doing ion dipole with the phosphate? Or what about this threonine here? Here is threonine 863. Is that threonine close enough to be doing ion dipole interactions? We found 1, 2, 3 possibilities, folks. They all seem to be in close proximity, and they're all neutral but polar, so they can do ion dipole. Lastly, we need to find one that does ionic. We went through most of the amino acids that are nearby. We got some non-polar ones, but mostly we have some polar amino acids. There's one in particular that's catching my eye that's nearby. This one right here. If you recognize the structure, you'll see there's a bunch of carbons and this blue thing. Blue is always going to represent nitrogen, just like how red is always going to represent oxygen in these types of structures. This is an NH3, which makes this lysine. Lysine is positively charged, and it seems to be in fairly good proximity to our phosphate molecules. So this seems like a pretty good candidate for ionic interactions. Let's click on it, and you'll see that this is lysine 866. Lastly, folks, in the last question I ask you, just have a little bit of fun. Be creative, do something cool with the structure. You'll see that I made my structure rainbow, I made my sticks in the middle purple. Just have some fun with it. You can do whatever you want to the structure. So, as an example, I'll do something else. First, I'm going to hide those sticks that I just showed. But now I want to have a little more fun with it. I think I'm actually going to make mine into spheres, because why not? Look at how silly that looks. It's kind of ridiculous. But I also want to make it color. So let's do a rainbow. I mean, I always choose rainbow. Otherwise, it's kind of boring. And maybe this could be the structure you submit to me. You can see some funky amino acids just hanging out. Or maybe, I don't know. Split pea. Oh, beautiful. Look at that beautiful split pea. Or maybe I want it hot pink. Oof. Now that's bright. So you can really do anything you want for this last section. Maybe I want to show my two substrates as sticks. I'm going to actually hide the spheres for those, and I'm going to color them, aquamarine. And bam, have you ever seen a more jarring picture? Maybe not. But that's really it, folks. For the last question, make a pretty picture. Try to have a little fun. And I hope you learned a couple things and explored a little bit in this program called PyMOL. PyMOL is very commonly used in biochemistry, chemistry, and biology. So it's good to kind of learn a couple new skills. I hope this was a bit enjoyable and provided a bit of review for glycolysis, since we got some time to look at hexokinase, its structure, and think about the mechanism and the binding of substrates.